Hello, everyone. Today we'll be discussing a question from October-November 2024, variant 1-1, and this is question number 6. Now, this question number 6 is of 6 marks in total, which is divided into 3 parts, that is 6A, 6B, and 6C, respectively. About this particular question, this is a question on the study that was conducted by Perry et al., and we come across this study when we're preparing for the social approach. Now, this fascinating study that was conducted by Perry et al. Examines how the hormone oxytocin influences our personal space preferences, especially in relation to our empathy levels. If we speak about the aim of the study real quick, the researchers were trying to investigate whether administering oxytocin affects individuals' preferred interpersonal distances and how this effect varies based on their empathy traits. About this study, this involved 56, there were 56 male undergraduates from the University of Haifa. They were aged between 19 to 32 years. The participants, they were first assessed for their empathy levels using the interpersonal reactivity index. Now since we're revising this, let's try learning whatever we can as I try helping you revise this. Like I said, the study involved 56 male undergraduates from the University of Haifa. You could get a question that might ask you about the characteristics of the sample. So you could very conveniently write there were 56, they were males, they were undergraduates, they were from the University of Haifa, they were from the age range from 19 to 32 years. Now, these participants, like I said, they were first assessed for their empathy levels using the interpersonal reactivity index, which was a 28-item questionnaire. Based on this scores, they were categorized into high or low empathy groups. Now, taking this ahead, if we talk about this study, the first question over here in the study by Perry et al., the participants had to complete 2 conditions 1 week apart. One was the oxytocin condition. So the participants had to go through 2 different conditions over here. The first condition over here was the oxytocin condition. Describe how the participants received the oxytocin. Now, this particular question, this is a 3-mark question, and we've been expected to describe how the participants received the oxytocin. So you see that the marks that are allocated to this particular question, there are 3 marks. And of course, out of the list that you all see in front of you, you're expected to write down any 3 correct points. Any 3 correct points you write, correct points you write would help you score a 3 on 3. Now, let's revise this real quick. The participants had to self-administer the oxytocin. There were 24 international units. That makes it around 250 milliliter. This was done in the presence of the experimenter. It was administered intranasal. This was done through the nose. This was done through a medical dropper. And there were 3 drops per nostril that were administered. So out of these points that you see, there are 6 written over here. Write down any 3 and you'd hit a 3 on 3 there. Moving on real quick to the second part of this question. The second part of the question over here reads, identify what was received by the participants in the other condition instead of oxytocin. Now, on one end, there was oxytocin, and on the other end, there was a saline solution, which I'm sure if you've done this study, this wouldn't be a difficult question for you. Just see that this question is, the command word used over here is identify. And when you've been asked to identify, and this question is of 1 marks, you can simply write down saline over there and get your mark right there. So we're done with the first 2 parts. Moving on to the third part of the question real quick. Explain one strength of this study. Explain one strength of this study. And this is a 2-mark question where we are expected to explain one strength of this study. Now, do not forget this. You get the first mark for identification of the strength, and you get the second mark for explaining the strength via example from the study. So you get the second mark for explaining the strength, and this should be via an example or through an example from the study. About this particular research, or rather, uh, if I if we say when we're conducting a research, it is important to make sure that the independent variable is truly responsible for changes in the dependent variable. You all would know this, where the independent variable is referred to as the IV and the dependent variable is referred to as the DV. A key strength of Perry et al. Study on personal space was the use of controls. Now, controls, this is a strength, the high amount of controls that were there by the researchers. So controls could be our strength over here. Now, these controls, they were used to ensure that oxytocin levels rather than other factors influence personal space preferences. Now, the question is, how is it that these controls were, how is it that the researchers ensured that these controls were used? One specific control in this study was the 45-minute waiting period after administering oxytocin or the placebo, that was the saline. This was done to allow oxytocin levels to plateau, ensuring that all participants had a similar physiological response before they were tested. Now, what we did over here was we identified a strength. Now, the question is, why is this a strength? This control was crucial because without it, differences in personal space preferences might not have been due to oxytocin levels, but rather variations in absorption time amongst participants. So, what we're trying to explain over here is that this 45-minute waiting period after administering either oxytocin or placebo, why is it that this was necessary? Now, as we standardize this, by standardizing the waiting period, the researchers could be confident that any changes in personal space were directly linked to oxytocin and empathy levels rather than any external factor. It could be like time inconsistencies or placebo effects, so on and so forth. Now, remember when this says 1 mark for explaining the strength via example from the study. Now, what sort of impact would this control have on the validity of the study? So, of course, if we're discussing this under the heading of strength, we should know the impact, the positive impact, and positive impact, of course, because we're using the word strength over here. Because of this strict control, the study's internal validity was strengthened, meaning that the researchers could be more confident stating that the IV, that is either the oxytocin and empathy levels, they were directly influencing the DV, that is personal space preferences, rather than any other uncontrolled variable. Now, quickly having a look at what the examiner has to offer us over here in the form of an example, there were controls in place to try to ensure it was the IV that was directly affecting the dependent variable, that is DV. For example, all participants waited 45 minutes to ensure OT, that is oxytocin levels, had plateaued, making sure it was the empathy or oxytocin affecting personal space. This is how we get the first mark. This is how we get the second mark. Now, you could use another example if you feel like. Again, the second example, there was a clear standardized procedure which improves reliability of the study, or the study can be replicated to check results, etc. Remember, there's a difference between the term replication and reliability. You refer to a research being replicable if the procedure of the study is standardized. If the procedure of the study is standardized, you can, of course, conduct that research all over again in exactly the same manner. So the ability to be able to redo a research in exactly the same manner, we refer to that as replication. However, when we use the word reliability, this is a step ahead. Reliability is when you're not only able to conduct a research in exactly the same manner again, but you also come up with the same results. So replication is repeating the same research again. Reliability is repeating the same research again and coming up with the same results or the findings. For example, in experiment one, the figure approached for 3 seconds in which the participants had to press the space bar to stop it getting any closer. Now, again, this is how you got your first mark through identification, and this is how you got your second mark uh, through explanation of that strength via an example from the study. Another third example that I'd want to discuss really quick, because of course you could receive 2 marks either by writing only this or by this or by this, but I'm just uh, doing this third example over here, third strength over here as well, because remember, you never know, the next evaluative question that you get right in the end, that is a 10-mark question, could be on Perry et al. So the more we know, the better it is. The double blind technique could reduce demand characteristics, which is again a strength. Now, what is a double blind design? When we use the word double blind design, a double blind design is when the participant as well as the person who's administering that particular part of the research, they're unaware of the conditions that they're a part of. So that is a double blind design. The double blind design or technique could reduce demand characteristics as the participants did not know if they had been given oxytocin or placebo. So this is how you get the first mark, and this is how you get the second mark. I hope if you come across a question of this sort, you know exactly how to score full marks. So this was question number 6. This was of 6 marks, and I hope you'd after this lesson you'd be able to score a 6 on 6. Hoping this was helpful. Thank you.